You know, um, if you are looking through a microscope at a standard slice of a cancer tumor to the untrained eye, anyway, all you see are these abstract pink and purple blobs just scattered across a glass slide.
Right, it just looks like modern art or something.
Exactly. But what if I told you that an artificial intelligence could look at that exact same, simple picture and instantly read the invisible underlying genetic code that's actually driving the disease?
It really is just a mind-bending premise.
It is. So welcome to the deep dive. Today, for you listening, we are exploring a massive data dilemma in the world of cancer pathology. And we're unpacking this crazy new AI framework called BiTro.
Which stands for bidirectional transfer learning.
Right, bidirectional transfer learning. And it's basically attempting to completely change the way we see those pink and purple blobs.
It is. We're looking at a fascinating research paper today, and what this technology represents is a potential paradigm shift in precision medicine.
Yeah, big time.
Because it's attempting to build a bridge across a very frustrating gap in how we currently understand, map, and ultimately how we treat cancer.
Okay, so let's unpack this for the listener.
Yeah.
Because to really grasp why BiTro is such a big deal, you have to understand the sheer mess that cancer data is currently in.
Oh, it is a mess.
Right. So let's use an analogy. Imagine you want to understand what's in this massive bowl of fruit. In the world of cancer research, one of our main tools for decades has been something called bulk transcriptomics.
Right.
But bulk transcriptomics is, well, it's basically like throwing that entire bowl of fruit into a high-powered blender.
You basically get a smoothie.
Exactly. You get a smoothie. Now, you can chemically analyze that smoothie
Yeah.
in staggering detail.
Yeah.
I mean, we are talking deep genetic sequencing here with like 60 to 150 million reads.
Which is incredible data. You know exactly how much fructose, vitamin C, fiber, all of it.
Right. You know everything that's in the glass, but, and this is a huge but, you have absolutely no idea what the original fruit salad looked like before you blended it.
Precisely. The structure is just gone.
Yeah, you don't know if the strawberries were sitting right next to the bananas or, if all the blueberries were clustered defensively on one side of the bowl. The physical arrangement, the architecture, it's completely erased.
Which brings us to the other side of the coin.
Right, because on the other hand, we have a much newer technology called spatial transcriptomics, or ST. And spatial transcriptomics is like looking at the intact fruit salad.
You see exactly where every single piece of fruit is located.
Exactly.
Yeah.
Which sounds ideal, right, until you look at the limitations of the technology itself.
Yeah, because mapping the genetics spatially is so new and, frankly, incredibly expensive that you only get a very shallow genetic read.
Shallow like how?
Well, you might only get maybe 25,000 reads per spot. Compare that to the hundreds of millions you get in the smoothie.
Oh, wow. That is a massive drop-off.
It really is. And worse, because of the sheer cost, you don't have very many bowls of fruit to actually look at. The sample sizes are tiny.
Which makes it really hard for a computer to learn from them, right?
Exactly. Small sample sizes are basically the enemy of machine learning.
And so this raises a huge question for oncologists. Like, how do you map the physical structure of a tumor, what they call the whole slide image, or WSI, to its deep gene expression?
Right, and that's the holy grail of cancer pathology right there. Because a tumor isn't just a uniform lump of identical bad cells.
It's more complicated than that.
Way more complicated. It's a highly structured, heterogeneous microenvironment. It has its own highways, its own supply line, its own defensive walls.
But until now, scientists have been stuck making this terrible choice.
Yeah. You either choose the deep but messy bulk data, the smoothie, or you choose the shallow but mapped spatial data, the intact fruit salad.
Because both data sets are, well, they're fundamentally flawed on their own.
They are.
Which brings us to the hero of our deep dive today, BiTro. This AI is stepping into the ring to solve this exact dilemma.
And it does it in such a fascinating way.
Yeah. And the first thing that jumped out at me in the paper is how it actually looks at the image itself. I mean, how does the AI even begin to process a whole slide image? Those hospital scans have to be absolutely massive.
Oh, they are gigabytes upon gigabytes of data.
So does it just like scan the whole thing top to bottom like a flatbed scanner?
If it tried to do that, it would overwhelm the computer's memory almost instantly. It's simply too much data to take in all at once.
So what's the workaround?
It has to break the problem down to the cellular level. The paper outlines this WSI pre-processing phase. First, it uses these specialized segmentation models.
Okay.
You might see names in the research like HoverNet or 𝗕𝗏𝗏.
Wait, what do those actually do in plain English?
Think of them as ultra-precise digital scissors.
Oh, I like that.
Right. They scan the massive image and literally find the boundaries of every single individual cell. And once they find a cell, they extract a tiny 48 by 48 pixel tile centered right on it.
Okay, so now, instead of one massive, unreadable image, the AI has millions of tiny, manageable pictures of individual cells.
Exactly. It's breaking the city down into individual houses.
That makes perfect sense.
Yeah.
So then what?
Then it feeds those tiny house images into a really powerful visual encoder called DynoV3.
DynoV3 sounds intense.
It's a vision transformer. And unlike older image scanners that just look at raw pixels left to right, a vision transformer looks at the whole tile and analyzes the relationships between different visual elements.
So it's looking for patterns.
Right. It mathematically defines the look of the cell, you know, how big the nucleus is, the texture of the outer membrane, the overall shape. It captures all these semantic visual features.
Okay, here's where I actually have to pause and push back a little bit.
Sure.
Why do we care so much about the specific look of one cell? I mean, isn't cancer fundamentally a genetic disease?
It is.
So why are we spending so much immense computing power having an AI stare at the physical shape of a single cell on a piece of glass?
That is the core question, isn't it? What's fascinating here is the deep biological connection between a cell's visual phenotype, how it looks, and its genotype, its underlying genetic expression.
Oh, okay, so they are linked.
Intimately linked. In biology, form follows function. A cell looks a certain way specifically because of the genes it is currently expressing.
Give me an example of that.
Well, a cell that's preparing to divide looks drastically different than a cell that's currently under attack by the immune system.
Right, of course.
So those visual clues are basically a physical manifestation of the invisible genetic code.
Okay, I see. So if the AI can deeply understand the shape, it can start to infer the genetics.
Precisely. But a cell does not exist in a vacuum.
Right.
Its behavior, its shape, and its genetics are heavily influenced by who it's sitting next to. And this is where BiTro introduces what the researchers call spatial enhancement.
Yes. I love this part of the paper. It uses something called graph attention networks to map out the neighborhood.
Yeah, think of a graph attention network like building a social media network, but for cells.
Okay, the cellular social network.
Exactly. The AI isn't just looking at the isolated cell anymore. It specifically looks at a cell's 8 nearest neighbors. It creates a mathematical graph of these local interactions and assigns what they call learnable weights to different neighbors.
Meaning it figures out who the cell is actually listening to.
That's a great way to put it. Because a tumor cell might be physically touching 5 other tumor cells, but it might be most heavily influenced by the one immune cell right next to it.
Oh, wow. So the network figures out those local dynamics.
It does. But then BiTro zooms out again. It uses a transformer encoder to look at the global long-range relationships across the entire tumor slide.
So it's looking at the whole map now.
Right. It adds positional encoding, literally GPS coordinates. So the AI knows exactly where every single cell sits on the map.
Okay, so just to summarize this pre-processing for the listener. We aren't just taking a picture of an isolated cell.
Yeah.
We are mapping its physical shape, its immediate social circle of 8 neighbors, and its exact location within the entire sprawling city of the tumor.
Exactly. It builds an incredibly rich, spatially aware profile for every single cell.
Which is incredible. But it brings us to the massive elephant in the room here.
The answer key problem.
Exactly. We have these amazing detailed profiles of individual cells.
Yeah.
But the AI's answer key is still completely flawed.
It is.
If the researchers want to train the AI to predict genetics, the only answer key they have to check its work against is the smoothie, the bulk data.
Right.
So how in the world does BiTro connect these millions of individual cell pictures back to a blended-up smoothie of genetic data? How does it know which specific cell contributed which specific gene?
That is the exact challenge. It's a classic problem in data science, actually. And to solve it, the researchers utilized a mechanism called multiple instance learning, or MIL.
Okay, let's translate that for the listener because multiple instance learning sounds incredibly dense.
It does. Think of it as a weekly supervised learning scenario. Imagine I hand you a large bag containing 100 different keys, and I tell you one of the keys in this bag will unlock a specific door.
Okay, I'm with you.
You don't know which individual key, the instance is the right one. You only know that the bag as a whole has the right key.
So the bag is the bulk data, the smoothie, and the individual keys are the single cells.
Precisely. The AI doesn't know the exact genetic label of the individual cell. It only knows the final aggregated label of the bulk smoothie.
So how does it narrow it down?
So multiple instance learning acts as a feature-level aggregator. The AI uses an attention mechanism to basically make highly educated guesses and verify them. It looks at the whole bag and says, based on the visual phenotypes and the social networks I've built, I calculate that this specific group of cells over here is responsible for this specific genetic signal in the final bulk readout.
But trying to figure out which key opens which door when you only have the smoothie seems almost impossible.
And if you only have one type of data, it practically is. And this is where BiTro's namesake, its real crown jewel, comes into play. Bidirectional transfer learning.
Yes. This is the part I was waiting for. How does it actually cross-train?
Well, the researchers realized something brilliant. The strengths of one flawed dataset perfectly cover the weaknesses of the other.
Okay, so the bulk data has massive sequencing depth, millions of reads, but no map.
Right. And the spatial data has the map, but a very shallow pool of reads. So BiTro uses an extremely efficient fine-tuning method called LoRA, or low-rank adaptation, to trade knowledge back and forth between the two.
Okay, let's break down LoRA really quick, because when I was reading the paper, I couldn't understand how a computer could constantly switch between learning from a map and learning from a smoothie without, like, completely forgetting the previous lesson or taking months to compute.
That is the beauty of LoRA. Imagine the AI's core visual model, the part that understands what cells look like, is a massive, towering library of information.
Okay.
If you try to rewrite every single book in the library every time you switch from spatial data to bulk data, it would take forever.
Right, the computational cost would be insane.
Exactly. So LoRA essentially freezes the massive library. It doesn't touch those books.
Oh, interesting.
Instead, it adds a tiny, lightweight sidecar of new trainable parameters. It's like putting a sticky note on the librarian's desk with specific instructions for the new task. It is incredibly fast and mathematically lightweight.
Wait, so it's basically using these lightweight sidecars to swap skill sets. Let me make sure I have this straight.
Go for it.
If BiTro is trying to predict the messy bulk data, the smoothie, it first studies the spatial data using a LoRA sidecar to learn how cells are naturally arranged.
Right.
And if it wants to predict the spatial data, it first studies the deep bulk data to learn the rich, complex genetic detail.
Yes, that's exactly it.
Yeah.
By pre-training the model on the spatial data, the AI learns the intrinsic rules of tissue architecture. Then, when you ask it to predict the bulk data, the AI already understands how cells spatially group together, which makes its multiple instance learning aggregations vastly more accurate.
It's like giving an architect the spatial blueprint of a house and then asking them to guess where the plumbing is hidden behind the walls.
That's a fantastic analogy.
They can make a much better guess than someone who just has a list of pipes.
Exactly. And the reverse is equally powerful. If you pre-train on the highly detailed bulk data, the AI learns the deep, complex correlations between different genes.
Meaning what, exactly?
It learns that if gene A is active, gene B is usually active too. Then, when you ask it to predict the shallow spatial data, it uses that deep genetic knowledge to fill in the missing blanks.
Oh, wow.
Yeah, the model ultimately converges on a highly accurate parameter space that neither dataset could ever reach on its own.
Okay, so in theory, this bidirectional cross-training sounds like an absolute magic bullet. But cancer isn't a theory, right? It's a messy, unpredictable, terrifying disease.
It is.
So when they actually tested BiTro on real patient data, did this mathematical trick actually hold up?
It did more than hold up. The researchers put BiTro through the ringer. They didn't just test this on one perfectly clean dataset. They tested it on 5 major cancer types from massive public databases like TCGA and the 𝗕𝗏 platform.
Wow, 5 different types.
Yeah, they looked at breast cancer, colon cancer, prostate cancer, liver cancer, and squamous cell carcinoma.
Which is huge, because for you listening, if you or a loved one is waiting on a biopsy result, the current standard of care often involves waiting days or even weeks for expensive, specialized genetic sequencing to come back from a lab.
Exactly.
If an AI can accurately infer those results just by looking at the standard tissue slide that is already sitting in the hospital, that changes the game.
It accelerates the timeline of precision medicine dramatically. And when it came to predicting that bulk transcriptomics data, BiTro went up against heavy-hitter baseline AI models. We're talking about models with names like 𝗖𝗏, 8-2RNA, and UNINA.
And how did it do?
BiTro, especially when using its bidirectional transfer learning, beat them out across the board.
Yeah, the paper mentions it achieved the best scores in Pearson correlation and Jensen-Shannon divergence. Now, I know Pearson correlation basically measures how well the AI's predicted genetic values line up in a straight line with the true values. Okay. But what is Jensen-Shannon divergence? That sounds like a sci-fi term.
It does sound like it. Simply put, Jensen-Shannon divergence measures the difference between 2 probability distributions.
Okay, so it's comparing shapes.
Basically, it's asking, does the overall shape and spread of the AI's predicted gene expression match the real-world shape and spread of the patient's actual tumor?
Oh, I see.
And a lower score means the AI's guess is much closer to reality. BiTro consistently achieved the lowest divergence scores.
And it wasn't just the bulk data either. On the spatial transcriptomics side, it went up against state-of-the-art models like histogene, His2ST, and M2AST. On the breast, liver, and squamous cell datasets, BiTro took the top 2 places across the board.
Which brings us to my favorite part of any complex AI paper, the ablation study.
I knew you would bring this up. For you listening, an ablation study is the part of the research where scientists intentionally turn off parts of the AI model to see what breaks.
It's my favorite part because it's so revealing.
It's like a mechanic taking parts out of a car engine one by one, right? Just to see which specific belt or spark plug actually makes the car stop running.
Exactly. It's the only real way to prove that your incredibly complex mathematical architecture is actually necessary and not just smoke and mirrors. And the ablation study for BiTro revealed a crucial finding about something called variance normalization.
Oh, right. The paper referred to this as dealing with the loud versus quiet genes, right?
Yes. In genetics, the dynamic range of expression varies wildly. You have certain marker genes that are expressed in the thousands. They are screaming.
The loud ones.
Right. And then you have vital transcription factors that might only be expressed in the single digits. They are whispering. And without variance normalization, the AI's learning process gets completely overwhelmed by the high variance loud genes.
It's like trying to listen to someone whisper a secret to you while you're standing in the front row of a heavy metal rock concert.
That is exactly what it's like. You literally can't process the quiet information.
So how does the AI fix that?
Well, mathematically, the loud genes dominate the loss function. The AI simply ignores the subtle, quiet genes, because fixing its errors on the loud genes yields a bigger mathematical reward.
It's chasing the easy points.
Exactly. By normalizing this variance, the researchers forced the AI to put on noise-canceling headphones. It forced the model to capture the diverse, balanced range of all gene expression patterns.
And that universally improved its accuracy.
Across the board.
That's fascinating. The ablation study also showed something really interesting about how the AI groups the cells. Early on, we talked about how it uses k-means clustering to group cells by their visual phenotypes, basically sorting them by what they look like.
Right, visually grouping them.
And the paper noted that this provided implicit regularization. Whoa, implicit regularization. Let's translate that for the listener really quick.
Implicit regularization is just a fancy way of saying it keeps the AI from overcomplicating things or basically memorizing the data.
Yeah.
By forcing the AI to group similar-looking cells together, it acts as a guardrail. It tells the AI, hey, these 10 cells all look identical and they sit right next to each other. They probably share a similar genetic function. Don't invent 10 wildly different genetic profiles for them.
Right. And the fascinating part to me was that when they turned this clustering feature off for the messy bulk data, the AI's performance actually dropped.
It desperately needed those visual guardrails to make sense of the smoothie.
But when they turned it off for the spatial data, it didn't really matter. The performance barely changed at all.
Which makes perfect logical sense if you think about it.
Really? How so?
Well, the spatial data already provides an explicit map. The AI already knows exactly where the cells are and how they're clustered, so adding a mathematical rule to group them by visual phenotype becomes redundant.
Oh, I see.
Yeah, it's a fantastic validation of how different types of data require different learning constraints.
Okay, so we've talked about predicting spots, and we've talked about predicting bulks. But I have to ask the ultimate question here.
Let's hear it.
The paper mentioned a term, pseudo-cellular spatial transcriptomics.
Yes.
Since BiTro is generating these incredibly detailed cell-by-cell visual profiles, is this model actually predicting the genetics of a single individual cell just by looking at it?
I want to be very precise here.
Okay.
The primary goal of BiTro was to map to the aggregated levels, the bulk smoothie or the spatial spot. However, as a byproduct of how it uses multiple instance learning, yes, it actually does generate pseudo single-cell profiles.
Wait, really?
Yeah. Because the AI has to mathematically justify its final answer, it basically calculates, I believe this exact cell right here contributed this specific fraction of the total gene expression.
And they actually tested those microscopic guesses, right? They didn't just take the AI's word for it.
Oh, no, they put it to the test against real super-resolution data. They use something called the 10x Genomics Xenium datasets.
What?
This is bleeding-edge technology that physically measures the transcriptomics of hundreds of thousands of individual cells. It is the ultimate answer key. And they pitted BiTro against dedicated single-cell AI models like iStar and GHIs.
Wait, models that were specifically built and trained for that exact single-cell task.
Exactly. And while a dedicated model like GHIs naturally performed best because, it was trained with explicit single-cell supervision, BiTro surprisingly held its own.
That's crazy.
It achieved highly comparable performance, which is staggering when you consider that BiTro was never explicitly trained on single cells.
It learned to infer single-cell genetics just by cross-training on the messy bulk and shallow spot data.
Exactly.
It figured out the exact shape and size of the individual fruits just by studying the smoothie and the blurry fruit salad. That is absolutely wild.
It speaks to the incredible power of bidirectional transfer learning.
Okay, we have covered a massive amount of ground today. Let's bring this all together for the listener. What BiTro has successfully done is build a computational bridge.
A very sturdy bridge.
All right, on one side of the river, we have standard, cheap, abundant tissue images. The kind of simple glass slides that pathologists have been looking at under microscopes for decades.
Yeah.
On the other side of the river, we have expensive, highly detailed, incredibly complex genetic sequencing. And BiTro proves that we can use AI to connect the two.
Right.
This means we can gain deeper, spatially aware insights into the tumor microenvironment without necessarily needing to spend millions of dollars and weeks of time on new deep spatial sequencing for every single patient.
Right.
We can unlock the invisible secrets hiding in the data we already have.
And that is the ultimate value here. In a world of absolute information overload, especially in the medical field, BiTro proves that we don't always need to invent a new way to gather new data.
Right.
Sometimes we just need a vastly smarter way to connect the data we already possess. By trading knowledge back and forth between flawed data sets, we can compensate for their individual weaknesses.
Like using the deep to illuminate the shallow.
Exactly. And the mapped to organize the messy, we quite literally make the whole greater than the sum of its parts.
I love that. A smarter way to connect.
Yeah.
Well, thank you for joining us on this deep dive. But before you go, I want to leave you with one final lingering thought that wasn't explicitly covered in the paper today.
Oh, what's that?
Well, if an AI can now accurately infer an invisible, complex genetic profile just by analyzing the visual arrangement of cells on a standard, everyday glass slide, what other invisible human traits, underlying conditions, or medical histories might artificial intelligence soon be able to see hiding in plain sight within our standard medical scans? Keep that in mind the next time you look into a microscope or a mirror. See you on the next deep dive.